This is my cell signaling and disease stop motion project, and I will be covering hepatitis C. Alright, so hepatitis C transmits through blood-to-blood contact. So, say someone infected with the virus somehow gets their blood in your open wound, you might now have hep C. Congrats. But what does this mean exactly? Well, the virus will now spread through your body, replicate, and cause your immune system to attack your liver cells, potentially causing some serious damage. I'm going to start by looking at this at a cellular level. The hepatitis C virus, or HCV for short, doesn't have a normal signal transduction pathway. It instead hijacks existing pathways within the host cells. In the interest of not boring you to death, I won't go into detail with every single pathway the virus hijacks. I'll just give you a quick rundown. HCV uses the RAS-EGFR pathway to counteract the antiviral JAK-STAT pathway and taking away its ability to fight infection. The JAK-STAT pathway is hijacked to suppress immune gene expression. HCV hijacks the cell's machinery in the MAPK pathways and manipulates it to replicate the virus. The transforming growth factor beta pathway is hijacked to essentially boost liver disease to full-blown liver cancer. The hedgehog pathway is hijacked to promote the process of forming scar tissue, which leads to organ dysfunction. I'm not going to try to say the name of these pathways, but HCV hijacks them to protect the infected cells from programmed cell death and to aid viral replication. The SREBP pathways are used to enhance lipid production necessary for viron assembly. The ROS-J and K-itch pathways aid in the release of new viral particles. Now I know that was not very straightforward, so here's how it actually works in application. The HCV itself is a single-stranded RNA virus, which is surrounded by a capsid inside of a lipid envelope made from the host cell membrane. Outside of the lipid envelope is a bunch of proteins, notably the E1 and E2 proteins. We'll come back to these in a second. The HCV targets cells in the host body called hepatocytes. They are the primary liver cells and make up almost 70% of your liver. Okay, back to E1 and E2. These proteins act as ligands and bind to the receptors CD81 and SRB1, which sit on the surface of hepatocytes. This triggers the HCV viron to be absorbed into the cell's endosome via endocytosis. Once in the endosome, the viral envelope fuses with the endosomal membrane and releases the RNA into the cytoplasm of the host cell, where it travels to the rough ER. The ribosomes along the surface of the rough ER translate the message held by the RNA, converting it into an HCV polyprotein. Enzymes that break down proteins and peptides, called proteases, specifically the NS23 protease and the NS3-4A serine protease, break the HCV polyprotein down into 10 viral proteins. A kinase inside the host cell, called casein kinase 1-alpha, phosphorylates NS5A, which switches HCV from replication mode to assembly mode. Now the HCV uses its core protein to build a capsid around the RNA by using the RNA as a scaffold. It also obtains a lipid envelope through interactions with the rough ER. The HCV is now once again a complete infectious particle. This particle now incorporates itself into very low density lipoprotein particles and is released from the host cell. Now the HCV has now successfully replicated itself. Each new viral particle that was made from this process will now spread and infect other liver cells. This constantly activates the body's immune system, leading to liver inflammation, cancer, and failure. That's all I have. Bye.